So we're gonna start today. Before we dive back into our last step of cellular respiration, by reminding ourselves of the steps that we've already been through and where they're happening relative to the cell at large and relative to the mitochondria. Take a couple of seconds to do this. We're not writing out a chemical equation. We're just interested in where we are. And we're about to dive into a step that kind of relies on location. Okay, so let's go through it. All right. So when we're starting out with a cell, we've got our big old cell with our cytoplasm inside, our cytosol. We got our mitochondria as one of our organelles. Okay. So far, after our first step, glycolysis, happening out here in the cytoplasm in the cytosol. Glycolysis, perfect. Glycolysis is out here. Okay. So then we have pyruvate as our carbon-containing molecule happening after we split apart the glucose in glycolysis. That's going to travel into our mitochondria. Okay, so that's our linking step. We're not labeling here. And then we know that the Krebs cycle and our oxidative phosphorylation are going to happen in the mitochondria. So I'm going to zoom in on that mitochondria right now. Okay. So we've got our vaguely kind of kidney bean-ish, I guess this one's a peanut, mitochondrion, right? So we know that we have an outer membrane, outer membrane. And if we have an outer one, we wouldn't bother specifying that unless we also had an inner membrane, right? It would just be the membrane. Right, so we're going to have an inner membrane. Oh, usually the foldy one. That inner membrane. Between the two, they're not like glued to each other. We got a space, right? So we have our intermembrane space in this gap, right? So this gap here is our intermembrane space. Okay. And then on the very inside, we have our mitochondrial matrix. Okay. So when we are thinking about the Krebs cycle, we should have happening here in the center in the matrix, the Krebs here. And the step we're about to talk about is going to have to do with movement, right? So we're going to have movement moving between areas. So the next step is going to involve the matrix, that inner membrane, and this intermembrane space. Okay? So when we think about oxidative phosphorylation, we have to understand all three of these pieces. So we've got space here and this matrix here, and they're separated by this inner membrane, although all of this stuff is still in the mitochondria. Right. So this was the last slide that we did on Friday, right? So thus far, when we're thinking about ATP, when we're thinking about energy, we're up to 4 ATP molecules made per glucose, right? So 2 on net in glycolysis. We have to spend 2 to get glycolysis started. We got 4, 2 total is our earnings for our Krebs cycle. Every time we run it, we get 1 ATP, but we run it twice per glucose molecule because we split the glucose in half in our first step in glycolysis. So that's how we get to 4. So the 2 net, 1 for the first turn of the Krebs cycle, 2 for the second turn of the Krebs cycle. So now we're going to get a lot of ATP in this final step, in this oxidative phosphorylation step. This is using this structure called the electron transport chain. So when we're thinking about the electron transport chain, what we're going to be thinking about how to have electrons, that's the name, moving down a series of proteins embedded in one of these locations we just talked about. We're going to be using them to synthesize ATP. So this is the step that's going to require oxygen. And didn't tell you the answer right there because I knew I was about to ask you, where is this electron transport taking place? So where are we located when we're thinking about that electron transport chain? Great. Yeah, so we're in the mitochondria. All right, so we're in the mitochondria, and we're interested in this inner membrane. So when we think about the electron transport chain itself, we're going to be thinking about the proteins embedded in this inner membrane. They're going to be passing the electrons. But the reason we're passing the electrons is that we're going to be trying to build up a gradient of hydrogen ions. So the purpose of this transport chain, the purpose of passing electrons, is going to be to force a bunch of hydrogen ions to build up in this intermembrane space. That's why we care about the parts, that's why we care about this space. So this is what that electron transport chain is doing. Okay. That's kind of step one of our oxidative phosphorylation. We don't number them, but that's the first part, what the electron transport chain is doing. And then at the very end of the electron transport chain, we're going to release that gradient. So when I say release that gradient, at the end, we're going to take all those hydrogen ions and we're going to let them push back through into the matrix, right? So here's our matrix. We had our inner membrane that we're crossing, and then we had the intermembrane space here. Right, so we're going to take those hydrogen ions, they're going to rush through like a fire hose into the matrix, and that movement is going to power the synthesis of ATP. So that's the actual making of the ATP there. That's what we're going to walk through in this next little bit, but that summary, you can draw that and explain that when it comes to exam time. That's basically what you need. Okay. So our electron transport chain is a series of molecules in an mitochondrial membrane. What they're doing is they're taking electrons that we've packed onto our electron carriers, right? Last time we talked about NAD and NADH, our electron carrier that we reduced in earlier steps, right? We took NAD, we added on, we gained electrons, and we could see that partially because we have that hydrogen on the end, but they're like a little visual indicator for ourselves. Okay, so we reduced them, we had this reduced electron carrier, and all of those reduced electron carriers were going into the mitochondria in order to set us up for this event. So it's use of the electron transport chain to do oxidative phosphorylation. Okay. So we reduced them earlier, and now we're going to do another series of oxidation reduction reactions where we pull off that hydrogen ion that's pushed into the intermembrane space, and the electrons we've just freed are going to travel down the electron transport chain. Okay. So overall, we're using this energy in the electron getting passed along to create our ATP. And this is the process of oxidative phosphorylation. We're going to name a step that, and we're also going to use oxidative phosphorylation as a word for one of our two ways we can phosphorylate our ATP. Remembering that phosphorylation is just tacking on an extra phosphate group. All right, so ADP, adenosine diphosphate, they're kind of low energy form of the molecule. It had two phosphate groups on it. ATP, which is what we want at the end of our cellular respiration, has three phosphate groups. So we phosphorylate it. This step is oxidative phosphorylation because we're using these oxidation reduction reactions. Oxidation reduction uses that oxidative phosphorylation there. We had substrate level phosphorylation in our earlier steps. And we do have an enzyme involved kind of at the end when we release this gradient of hydrogen ions or protons, okay, that we're about to produce. And we're going to see that this is the step in which we actually use oxygen. So this whole time we've been talking about how cells make ATP in the presence of oxygen, so aerobic cellular respiration. We didn't actually see oxygen being used until right now. The oxygen comes in right at the end for our final step. So here's some chemistry showing us what's going on with those electrons. So you can see, right, our reduced electron carrier that we built up. Right, most of it is NADH. We do have some FADH2 from the Krebs cycle. Right, the Krebs cycle It's a little bit of ATP, 2 per glucose molecule and 1 per cycle. But the big thing that the Krebs cycle is doing for us was reducing a lot of these electron carriers, giving us lots of NADH plus FADH2, less common, but still an electron carrier. And now as we pass those electrons into the chain, we're reversing it. So we're going back to our precursor molecules, our oxidized forms, the NAD plus and FAD, peeling off those hydrogen ions and peeling off electrons. Okay. So we peel off these hydrogen ions move into the intermembrane space. The electrons have a kind of domino effect where a series of reactions basically uses up their energy. When we're done at the end, we need to do something with those electrons to kind of be finished so that all our proteins in that inner membrane can kind of reset. So at the very end of this process, we have some kind of low energy electrons that we take, combine with some hydrogen and our oxygen. Okay. This is the only oxygen use in our aerobic cellular respiration, but it's super important. Gives us our H2O. That's why it's a byproduct at the end of it's a product at the end of our cellular respiration. But critically, what we have happening as part of this step, right, is we've gone from NADH to NAD plus as well. So this lets us kind of go back to the beginning and do it all over again, right? So oxygen is only actively being used in this step as part of the electron transport chain and our oxidative phosphorylation. But if you don't do this, if you don't have oxygen, you don't make water, and you don't use oxygen as a final electron acceptor, you basically run out of NAD, right? You run out of NAD, you run out of FAD in the cell, and so you just can't do metabolism anymore. You can't do this anymore if you run out of those molecules. So we have to reset everything. So here we have a kind of diagram of what's happening. So we got our location. We're zooming in the mitochondria. We're zooming in on that inner membrane. And we're noticing that we're looking at the intermembrane space on one side. We're looking at the matrix on the other side. And we're thinking about the relationship between these two locations, and we're thinking about movement. So step by step. Here we have those proteins or coenzymes that are embedded in that inner membrane. We're not going to learn their names or focus on them, but there are a series of them. Right? So what we have happening is we have our reduced electron carrier, in this example NADH, coming into contact with this first part of the electron transport chain, peeling off those hydrogens, peeling off electrons, and resetting to that oxidized form of the NAD+. Next, we travel down the chain, down the chain, down the chain. As we do it, we're taking hydrogen ions and pushing them through into the intermembrane space. I say push because you want to think about this like packing a crowded train or like packing your suitcase full of stuff when it's already full of clothes, right? It takes energy, that's what the electrons are for, to pack stuff tighter together, right? So we're packing all these hydrogen ions tight together, lots and lots, rather than letting them just kind of spread around in the matrix. For the moment, we have a relatively low concentration, low number of hydrogen ions. So we refer to this as like building up our proton gradient because a hydrogen ion is just a proton. So same thing regardless of how it's described. Okay, so we do that going down our electron transport chain. And then at the end, once we're done using the energy in that electron, we allow oxygen to combine with a couple of hydrogen ions, and we get our water, our product. Okay. So once we've done this, now it's time to use this buildup of hydrogen ions. So to use this buildup of hydrogen ions, we have to allow movement. I think of this like more like a wind turbine making energy or like a water wheel, right? This is what's happening as we allow these hydrogen ions to flow through ATP synthase, this enzyme at the end. So as we allow those hydrogen ions to flow through, they phosphorylate our ADP. So phosphorylate meaning they just add on a phosphate ion. So here's our adenosine diphosphate with its 2 phosphate groups. Here's our free floating phosphate ion, just happens to be in here. There's plenty of it in the matrix. And by releasing this energy in the form of movement of hydrogen ions, we make ATP. And once we do it, we would see this gradient, so this difference in the number of hydrogen ions on either side of the membrane. And we set it to equal, so constant equilibrium, before we build it back up again and do this. So I'm going to review all our steps one more time. So remind me which part of our cellular respiration happening outside of the mitochondria didn't require oxygen. Yeah, so the glycolysis was out in the cytosol, just in the cell generally. The other 2 steps are happening in the mitochondria. Krebs cycle is fully in the matrix. And then this oxidative phosphorylation is about movement happening across that inner membrane, so from the intramembrane space to the matrix. Okay. So going through our whole diagram in full of what we've covered. Right, we start with our glucose molecule, C6H12O6. During glycolysis, we used some NAD, so our oxidized form of our electron carrier, and reduced it, giving us some NADH. That's going to get shuttled into the mitochondria to be used as part of the electron transport chain and oxidative phosphorylation. And on net, we made 2 ATP through substrate level phosphorylation, meaning we used an enzyme to take adenosine diphosphate and tack on phosphate ions. So we used an enzyme, active site, bind in these things with the substrate, make this reaction happen. So next we had our linking step. So we take our product of glycolysis, which is called pyruvate. So that's where our carbons are at this point. So glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule. We just split our glucose in half. So we take that pyruvate and we shuttle it into the mitochondria. Okay. We do change our pyruvate molecule along the way. Okay. So it turns into acetyl-CoA, the end of this linking step. And as we're breaking down the pyruvate, we made a little bit more of our reduced electron carrier. So we can see some NAD turning it to NADH. And we peeled off some carbon dioxide. So we go from a 3-carbon molecule to a 2-carbon, and we can breathe out that CO2. Our next main step that we are considering for this class is our numbers of steps, is our Krebs cycle. So our acetyl-CoA goes in, we make oxaloacetate, not listed here, and then we have lots of steps that bring us back to the beginning, producing oxaloacetate again. And along the way, we notice that we make tons, relatively, of reduced electron carriers, right? So each of these other pieces only made 2 apiece, so we only had 4 at this point. Now we're making a lot more. So 8 of these reduced electron carriers coming from the Krebs cycle. We get rid of the rest of our carbon dioxide that we're breathing out. And per glucose molecule is how we have this numbered. So per glucose molecule, we made 2 ATP as part of the step. We got a little more ATP, cellular energy, still through substrate level phosphorylation. Final step is oxidative phosphorylation. This is where we make most of our ATP, 30-ish molecules of ATP. We got 2 in glycolysis, 2 total for Krebs cycle. Now we got 28 in oxidative phosphorylation. We're doing this by using those reduced electron carriers. So you can see now we don't have the H's on the end anymore. We peeled off the electrons, we peeled off the hydrogen ions. So we're going back to NAD and FAD. And we've used oxygen to accept those electrons at the end of the process, tacking on some hydrogen ions. We peeled off lots of them, right? Giving us H2O, giving us our water as one of our products in secretion, in addition to our ATP. So we now have the energy that was in the bonds of glucose trapped in this nice little ATP molecule that we can use generally in the cell for energy. We know this process isn't perfect. We know that we also lost some energy as heat here. That depends on kind of how effective the mitochondria are. That could be a little variable, but none of them are trapping 100% of the energy. That's fine. We like to have a temperature that's like higher than the air. So not a bad thing overall. So here we have that overall reaction. When we see this reaction right now, it's usually glucose, 1 glucose plus 6 oxygen molecules, gives you 6 carbon dioxide molecules plus 6 water molecules. We usually don't write on the ADP and the ATP, but they're there. That's where we trapped the energy. And that's our big process. I'm going to do a little bit more vocabulary reminder or new vocabulary. So when I say something is a catabolic reaction, what do I mean by that? What is a catabolic reaction? Cool. Yeah, so catabolic reaction release energy by breaking down a molecule. So when we say that we break a covalent bond and release that energy, that's catabolic. The reverse of this would be anabolic. So anabolic reactions build up molecules. Remember this silly way. I have a friend named Anna. She's pretty tall. I pretend she has a little cat, and I picture Anna and her cat every time I see these words. So, I was asking you or reminding you of this word catabolic because our next slide is titled glucose catabolism in absence of oxygen. So putting those together, we're talking about glucose breakdown, breaking down glucose, getting its energy when we don't have oxygen. So that can happen for a number of reasons, right? We could think about like other species living in low oxygen environments, but in our human bodies, we're going to have cells that are in low oxygen environments as well. So we're going to think about when you're doing a lot of activity, right? So say you're weightlifting or something, you're using up all of your energy really fast, you're using up your oxygen fast to feed your muscle. You can have low oxygen environments in your body, right? Temporarily or potentially longer term, depending how long you're exercising. You still want to have energy then, right? You don't have any energy, your cells can't do anything. So that's no good. So we have these processes where we can still break down glucose. We can still get the energy out of the glucose, but we don't have to have oxygen, right? So we can still get some amount of energy out of our carbohydrates without oxygen. And that's why we were interested in which steps use oxygen and where they're happening. Okay? So in the case where we don't have oxygen or low amounts of oxygen, let's say, we're essentially going to get the electron transport chain backing up. It's going to be full. It can't do that final step of letting the final low energy electrons turn into water using our oxygen that we fed in. So we have nowhere to oxidize those reduced electron carriers. We can't pull off the hydrogen ions. We can't use the electrons. We can't build up this hydrogen ion gradient anymore. So this the Krebs cycle, which was making those reduced electron carriers, is also going to stop, right? We're running out of NAD to turn into NADH. So this is a problem because our oxidative phosphorylation is happening in the mitochondria. We were using those reduced electron carriers for this process. That's how we were making most of our ATP in the cell. But we can keep getting a little bit of energy from glycolysis. So that's why it's important that glycolysis was happening outside of the mitochondria. So if your mitochondria aren't working right, it doesn't matter for glycolysis. And so we've got a different location. You don't need a mitochondria for it. But we can only continue to do glycolysis and get our 2 ATP per molecule of glucose if we keep oxidizing NADH. So we still are going to run into the problem of running out of NAD. We still are going to make NADH in glycolysis regardless of if it has anywhere to go, right? Even if it doesn't go into the mitochondria and power oxidative phosphorylation, that's just how the chemical reaction works, right? It's one of the products of those intermediate steps. So we do have to still have a way to turn NADH back into NAD. So we can keep doing glycolysis, keep getting 2 ATP, 2 ATP, 2 ATP per molecule of glucose. This is where lactate comes in. So when you hear like lactic acid, when we think about the synthesis of lactate, we're thinking about anaerobic respiration. So anaerobic, meaning in the absence of oxygen. Sometimes we call this fermentation. We're doing a slightly different process at the end in order to make sure that that NADH turns back into NAD so we can keep doing it. So that's where the lactate comes in. Right, so most of this runs as normal. Right, so in the absence of oxygen, we still take our glucose, we still break it down and turn it into pyruvate. Right, we still make 2 ATP, and we still make NADH. Right, so none of this changes yet. Okay, so all of that runs the same, glycolysis runs the same, but our mitochondria are not working as normal. We're not passing pyruvate into the mitochondria. We're not running the Krebs cycle. We're not running the electron transport chain to do oxidative phosphorylation. So in order to keep this going, to keep making NADH, which is just one of the steps of glycolysis, we take this pyruvate and we convert it into lactate. Right. So part of just how this reaction works, when we take pyruvate and use this enzyme lactate dehydrogenase, I'm going to expect you to remember the full name, but remember that ACE at the end is an enzyme, right? Lactate dehydrogenase. Right? So we take pyruvate, use lactate dehydrogenase, our enzyme, to catalyze a reaction where we get NH plus and lactate out at the end, basically. So if for this reaction, this lactate step, conversion of pyruvate to lactate, we're looking at the pieces, right? We take pyruvate and NADH as our reactants, and then we're producing our NAD plus and lactate as our products. And the whole purpose of this is to get that NAD plus so that we can keep doing glycolysis, right? That's our goal, right? Our goal is to keep running glycolysis because that's where we get ATP from in this process. And this is just to make sure that we're setting up our reactants for glycolysis again. So when we think about like lactic acid buildup, which is sometimes implicated in like maybe why muscles or tissues kind of burn or even exercising, but whether that's exactly true, we'll talk about later. But when we talk about that, we're talking about this process, right? We're talking about you used up all your oxygen because you were exercising a bunch. You still need energy. You're still going. You're not breathing fast enough to get enough oxygen to all your cells. So your cells are switching to glycolysis, still getting a little bit of ATP so they can still function, even though you're not getting them enough oxygen. You guys can take a look at this little, yeah, we'll watch it. It's just a quick little sort of video so we can see some of these things in a little more 3D rather than the me drawing arrows.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Okay, so we'll talk about digestion and absorption way in the future in A&P 2. For right now, what we gotta know is about that cellular respiration process, so how we're making the ATP. We know a little squidgy on the numbers, especially at the end of that oxidative phosphorylation step, in part because different tissues and different species have different effectiveness of that process, but we're making sure that we know 2 ATP from glycolysis, 2 from the Krebs cycle per molecule of glucose. Those things stay the same. So next, we're going to think a little bit about how we store energy and also how we can use things that aren't glucose for energy. So it's not the case that you only eat glucose, you only eat that little monomer, and that's it. That's the only thing your body can use. We also eat plenty of other types of macromolecules, plenty of other types of carbohydrates, and we can convert them all into energy. We, in general, use the same parts of this process. We just kind of jump in and out of this metabolic flow at different spots, depending what we're breaking down. So we'll see that some of the things that we're going to be using for energy, in particular, one of the important ones for us to know about is going to be glycogen. So glycogen is a storage molecule for glucose, and I'll talk about it several times throughout this course. Okay, so glycogen is a larger carbohydrate that we can get glucose out of and then run the whole process. You could also break down proteins for energy. Right, so in the case of proteins, we break them down into constituent amino acids. So we pull apart those amino acids to make our polypeptide chain, and we're then folded into a protein. And depending exactly what they are, we're going to jump into the process somewhere after glycolysis. So we're going to jump in somewhere here on this pyruvate step and into the Krebs cycle with our proteins. And then our triglycerides, also really important. So triglycerides, fats, right? So lipid, type of fat, or sorry, a type of lipid, and inert fat, right? Fat is for kind of long-term storage of energy. We know that from our daily lives. But from our chemical perspective, when we break down a triglyceride, we're going to pull apart the fatty acids from the glycerol backbone that's holding them together. So we saw that our triglycerides had that kind of flag shape to them, glycerol backbone, and then the tri is for the 3 fatty acid tails on here, right? So a little different than a phospholipid, but also a little similar. Okay. So our glycerol is essentially going to work like a carbohydrate, so that's going to go into the glycolysis step. But those fatty acids, like the proteins, jump in later. So the fatty acids are going to be jumping in here around the Krebs cycle and into the electron transport chain. And that process is going to be pretty good at producing energy in particular. Fats are a good way to store energy long term. Okay. So we're going to think about the glycogen first. Okay. So glycogen, we have a picture of it down here. It's made up of multiple different glucose subunits. So each of these pieces is going to be a glucose. Right? So you can see that it's this kind of branched chain structure. Starch from plants is similar, but not so branched. So glycogen is a storage molecule that we're going to have in liver and in kidney cells in particular. Okay. So in our liver and our kidney, these glycogen stores, we're going to be able to break apart our glycogen, pull off these glucose units, and put them into the bloodstream so that we have glucose for other cells to use. We're thinking about liver and kidney in particular, because while other cells will have some glycogen, not all the other cells are going to be able to convert it into glucose that can leave the cell. Okay, so the liver and kidney are special because they have an enzyme called glucose 6-phosphatase that converts the freshly peeled off glucose, which is in the form of glucose 6-phosphate, to like normal glucose that can travel across cell membranes. So when we think about making new glucose, we're going to call it gluconeogenesis. This is going to be a similar process. We'll pull together a bunch of glucose type words together on the next slide, I think the next one. So we also have this process of glucose for glucose, neo for new, genesis for creation. So when we say gluconeogenesis, making new glucose, we're focused on the liver. When we make new glucose, rather than peeling it off of glycogen like we did in the previous slide, of glycogenolysis. When we make new glucose, we make it out of glycerol, we make it out of lactate, and we make it out of amino acids. It's mostly happening in the liver. It's not exactly reversing glycolysis. There are different enzymes involved, but it's a little similar to doing the reverse of glycolysis. What we want to know is that we can take things that are not glucose, that are not carbohydrates, and turn them into glucose in this gluconeogenesis process. So here are two of the important steps that would be involved. In case you're curious and want to dive into the chemistry. Okay, here's the math. So what's happening here? Controls. Okay, so here we can see how we can make glycogen, how we can break down glycogen, and how we can make new glucose. Okay, so when we see a word that starts with GL or GLY, we want to be careful that we read the whole word and pay attention to the end of it. Okay, so the process that we talked about first, happening in the liver and kidney, breaking down glycogen to get glucose off. That's the glycogenolysis. So you peel out the glycogen for glycogen, lysis for breaking apart. So this is where we have glycogen. Peel off those little subunits. They're initially going to be glucose 6-phosphate. We convert them into glucose that can then leave. We can reverse processes using things like pyruvate to store things as glycogen in glycogenesis, right? So making glycogen. We saw on the previous slide that we can have glyconeogenesis or making glucose. So we'll stop there and I'll talk about fats and maybe a little more about this Wednesday.